me get back to welcoming everybody. Our conference is actually supported in large part by a generous grant from the National Institute on Aging, and we really appreciate their support. We also have other sponsors, as you can see up here. This is a joint program of other foundations, the Michael J. Fox Foundation, and Multiple Sclerosis Society, Fast Forward, and Forest Laboratories, JSW, and BioFocus. I'd like to thank our Scientific Advisory Committee, who worked hard to put the program together, and also our staff, who has done all the work to bring us here today. There is a survey that we'll ask you to fill out so we can make the program better next year. And this conference will be webcast at the end. After a little while, once we put the webcast together, you'll be able to access all the speakers on the internet. And there is CME available. Finally, we'll be having our 13th International Conference on Alzheimer's Drug Discovery. That's the view from the deck of the conference room where this conference will be held just across the harbor in Jersey City, and that'll be in September. We'll have about 2.5 days of about 30 presentations of very specific drug discovery programs that we funded or that are related to our programs. So it'll be 30 novel targets and drugs that'll be discussed there. And sort of the obvious goals I just want to say are that we're trying to focus this meeting on talking about the principles and practice of drug discovery with specific focus on the unique aspects of drug discovery for neurodegeneration. I'm told we have about 200 attendees here from 20 countries, about 40% people from industry and the remainder from academia and government and other foundations. And there'll be ample opportunities to create alliances and talk. We'll be having a cocktail reception after this session upstairs, and there'll be receptions during the course of the meeting. So neurodegenerative diseases, which we'll be talking about in the next 2 days, affect over 22 million people worldwide. And the WHO estimates that neurodegenerative disorders will be the major unmet medical need of the 21st century, surpassing cancer as the world's second leading cause of death in the coming decades. And as you can see here, most of these will come from patients with Alzheimer's disease, Parkinson's disease, ALS, multiple sclerosis, and Huntington's disease, all of which is actually going to be represented here in the next couple of days. And the unmet medical need here is that we have, for most of these we have, but not all, we have some symptomatic agents, but we really don't have any disease-modifying agents, and for those where we do have some disease-modifying agents, they're only really marginally effective in slowing down the course, but certainly not halting the course, which would be the most clinically meaningful outcome of a drug treatment in a neurodegenerative disease. Now, I know many of you probably know this figure, but for those of you who don't and are new to drug discovery, many of you are biologists who don't know much, and so we're going to try to start from a high-level view. But certainly, basic research would be sort of off the scale here to your left, and that's where targets are discovered, new pathways. But that's not really what leads to drugs. And what I would like to say is that the principles and practice of basic scientific research, basic neurobiology, are incredibly different from the science of drug discovery, where we need medicinal chemistry, pharmacology, and other disciplines to actually create a drug, which is actually an incredibly hard thing to do. And especially for neurodegenerative diseases, which are complex and multifactorial in origin. And Dr. Lipinski, who will be following me, will be drilling down on many of the points I'm going to make in the next couple of slides. But in a program, depending on how you go about it, you might start off with 10,000 to 1 million compounds, and that might go into a high-throughput screening campaign. A lot of biology and chemistry, but in this case, the biology is really looking at the effect of drugs on cells and trying to refine a molecule. But this is really the point at which innovation comes from in terms of new drugs. And so it's really a critical point, often unfunded, in the process of moving drugs forward into the pipeline. Obviously, the next stage would be animal studies, and then pharmacology, safety and toxicology studies. Once proof of mechanism has been identified and a lead optimized, and then an IND enabling process begins where drugs are prepared for human use. The IND is submitted, and then it goes into clinical trials. And we're not going to talk about clinical trials during the next couple of days, but you can see that this process generally is thought to take something like 12 to 15 years and cost something over $1 billion. I just want to say, though, that, for example, in Alzheimer's disease, the cost of one phase 3 clinical trial for a disease-modifying therapy is about $300 to $400 million. And you need about 2 of those to get an FDA approval. So when we talk about $1.2 billion, a lot of those costs are way out here. But a campaign for an initial innovation on new drugs can be a few $100,000, and certainly something manageable to get a program going, look to see if there's interesting leads. And obviously, as things go forward, things become more expensive. Now, just as a high-level perspective, estimated to be 20,000 human genes and 100,000 proteins made by these genes on the biology side. Many of these proteins are not accessible to drugs. Many are intracellular, and not really, they might not have hydrophobic pockets. And so many of these proteins are not biologically accessible for drugs, even though many of us think that proteins are of great interest for molecular pathways in biology. At the same time, if you look at the universe of chemicals that would be out there, it's just extraordinarily huge. 50 million compounds in chem abstracts, and estimated to be potential for 10 to the 40th to 10 to 100 possible small molecules. When you take that opportunity and you consider that there's been about 10,000 drugs approved by the FDA in the last half century or so, that seems like a big number. But most of these are actually historically been variants on formulation and delivery, me-toos, and that sort of thing, which isn't really possible anymore in the current environment. The cost effectiveness and the marginal benefits of tweaking a molecule and getting new intellectual property are not really acceptable as it was decades ago as a business model for industry or for our societies. And also many of these 10,000 molecules or approved drugs were antimicrobials, not drugs for chronic diseases, and certainly not drugs for neurodegenerative diseases. And if you drill down a little further than that, it's quite remarkable that there actually been approved less than 500 distinct chemical entities out of this universe, targeting only about 266. That's an estimate from a recent article, targeting about 266 human genome-derived proteins. So despite a huge opportunity on the chemistry side and on the biology side and on targets, I think this illustrates both the opportunity for diversity but also the difficulty in actually getting a drug. And if you look at small molecules, among the approved drugs, these are based on less than 50 unique chemical scaffolds. So a hard thing to do. Now, as biologists in neurodegenerative disease, I think we all, regardless of which one of the ones which are the topic of our meeting, we're dealing with some common pathways here, and so it might be possible that a drug that could be good for Parkinson's disease, for example, might also be good for Alzheimer's disease or chronic multiple sclerosis. And so these are the hypotheses that you all know about: oxidative stress, inflammation, misfolding, diseases of aging, mitochondrial dysfunction. And I think many of us as neurobiologists, we think about targets in this sense, and we find a new pathway and we think that's great, and we're going to go on a drug campaign. But as it turns out, the drug discoverers, if you will, don't really think about things the same way. We often get grants about ideas where biologists want to inhibit protein-protein interactions or go after a kinase, which is a rather difficult drug target because of the promiscuous nature of kinases and the difficulty in trying to get specificity and engineer out safety issues and so on. So there's a lot to learn here, let's put it that way, for a biologist about what is really a druggable target. And I hope that we'll be able to teach or convey some of that over the next few days. And the other thing is that biology is incredibly complex. Mother Nature and evolution has evolved enormous redundancy in our cells. And I know you can't see any of this, and I did put it up purposely just because this is a simplified cartoon of some of the biology at the synapse. And you can imagine that if, for example, we've seen some drug programs, both in industry and academia, which attempt to target GSK3 beta as a drug target. Well, you can see that the cell could get around that pretty easily through other pathways if you try to focus, with a high potency drug on this one target. That efficacy might be hard to get at just by inhibiting this one path, this one piece of a very complex biological network. And at the same time, when you try to go for a high potency interaction here, the potential for side effects increases. So thinking about trying to develop drugs for chronic diseases with multifactorial origins from a systems biology point of view, I think is a new concept that could be helpful. And one of the ways to get at that is something that Dr. Lipinski is going to tell us more about in a few minutes, which is that we typically think about target-based screening. You get your enzyme, you figure out the pocket, and you try to engineer a molecule by medicinal chemistry that hits that target. But there was this recent review in Nature Reviews Drug Discovery, as in one example, pointing out that if you look at the follower drugs, the Me Too's, the ones that I talked about a little earlier, that these would be drugs that are tweaking an existing known drug that actually works. You can see that target-based screening with medicinal chemistry refinement accounted for most of those successes. But phenotypic screening, which is actually what very often biologists do in cell culture, to look at an outcome like increasing ApoE in a cell culture, for example, on a screening campaign, to look for molecules where it's kind of looking at a black box and saying, okay, without knowing the actual molecular mechanism of action, can we find molecules that do what we want to do, decrease beta amyloid, increase ApoE? And you can see that actually there's been quite a few successes. In fact, the majority of first-in-class drugs in the recent past have actually come from phenotypic screening. And in the field of Alzheimer's, a good example of the differences here are how gamma secretase inhibitors were developed, which was on a phenotypic screening basis because the nature of gamma secretase wasn't really defined. There was no molecular target that could be crystallized and actually, a molecule actually designed. So there was assay development, high throughput screening, identification of hits, and that whole process before one got down to medicinal chemistry, whereas the beta secretase inhibitors were really done based on rational design, since we knew that this was an aspartyl protease, and the beta secretase involved in Alzheimer's disease was crystallized, the structure became known, the pocket was seen, it could go up into computational programs, and medicinal chemistry could be derived from that. So two different ways to develop a drug. So let me just close with the last few couple of slides about a recent trend which we've seen about increasing access to drug discovery in academia. A lot of really good drugs have actually come out of academia, and we see any number of buildings on campuses that are named after the drug discoverer that actually brought in $100 million to the university through royalties on licenses that was based on a drug, intellectual property of a drug that was developed at university. So we've seen a lot of successes, and it is possible with robotics and things and collaborations that drug discovery can be a source of innovation that builds on the basic research that academics are so used to and so good at. It does require a different way of doing business with a lot of resources and collaborations between teams of investigators and partnerships, and we're seeing more and more partnerships. Pfizer, Lilly, many of the other big pharma are now setting up various kinds of collaborations with, with academic centers around the United States and in Europe and elsewhere to try to bring the rigor and the expertise and the resources of big pharma to the academic investigators who, in general, don't have a lot of experience and not all the resources to conduct drug discovery. Certainly seeing a role for biotechs, although the funding environment right now is quite difficult. Nonprofits like ourselves are getting involved, and government, we're seeing more and more government involvement in trying to develop programs to fund drug discovery in the various disease states, and particularly in neurodegenerative disease. So that's all good news. What really needs to be done in academia to get this done, though, just as an illustration, again, and an introduction to our meeting, is that many of us here in the room who are basic neurobiologists are quite interested in target, basically what we call target identification. In other words, identifying a pathway that might be relevant to, for example, Alzheimer's disease, and getting that information out there. But that's really only the first step. And so we talked a few minutes ago about phenotypic screening, high-throughput screening, structure-based chemistry, lead identification, and optimization. Well, you need stuff to do this. You need assay development, chemical libraries, computational chemistry, and you need people that really deeply understand medicinal chemistry. They're the wizards that see molecules in their mind. They understand synthesis. They can put groups on that change the nature of a molecule so it becomes more druggable. And to me, you know, watching the wizards of medicinal chemistry work is quite amazing. We also need pharmacologists to be able to tell you if something is druggable and do the PK/PD, which comes when we go to in vivo testing and preclinical proof of mechanism in animals. And for these, I really believe that we need people that are expert in animal model testing, because our foundation and the guidelines are out on the desk there that we just published through an expert panel, kind of addressing some of the really serious issues in how animal models are conducted and reported, and often not blinded, not randomized, subjects, not enough subjects per group, and all sorts of problems, and a lot of data reported in the literature that's hard to reproduce or is not of high quality, and a lot of negative publication bias where negative reports are not published. So a lot of problems here, and we need experts in that. And of course, this is the point at which biomarker development becomes so critically important, because biomarkers are really the segue or the interface between preclinical development and clinical development. You know, the mantra, the holy grail is to find a translatable biomarker, one where you can test in animals, you get an effect, and then you can go into phase 1 and phase 2 and see if your drug actually works in humans. And then after that, we have, I mentioned the IND enabling and the ADMET formulation and scale chemistry. And again, this requires another kind of person, another part of the team, the pharmaceutical scientist, to do this work, and then finally the clinical trialist. So, building these teams in either virtually through collaboration or at your medical center is a critical thing in terms of conducting the drug discovery that you want to do. Just a brief mention of ADDF. For those of you who don't know, we've been in business about 13 years now. We've given out about $55 million to about 370 Alzheimer drug programs in 20 countries. We've funded 55 biotech companies in their early stages. We've seen something like 2,500 new ideas for drugs for Alzheimer's disease over the last 13 years or so, and as a measure of success, the programs that we funded have been able to garner over $2 billion in follow-on commitments, both in industry and in government grants and so on. And we've been able to move several programs that started really with just target identification are now finally in the clinic in phase 2. So we're proud of that, and we're happy to help, and we're here to give away money. That's our job. And we're here to help. We have 3 PhD scientists that are on our scientific staff, not including myself, and we want to help you. We can provide consultation and networking, at least in the field of Alzheimer's disease, and that's our website, and we'll all be here at the meeting. So let me close by saying that this drug discovery thing is often thought of as the valley of death, but we like to think of it as an amazing journey. Thank you very much. And it's a great privilege now to introduce Dr. Chris Lipinski. Dr. Lipinski spent several decades, I would say, 30-some-odd years, 32 years, in industry discovering drugs, so he knows the whole thing quite well, and for the last few years he's been on his own consulting and advising companies and biotechs and serving on committees at the American Chemical Society and doing all sorts of great things and trying to advance the field of drug discovery. And of course, he's most famous for the Lipinski Rule of 5, which is used all over the world to identify molecules that might be druggable. So.
Thank you, Howard. So am I using this as opposed to the... No, don't use this?
You don't need it.
Don't need it, okay. Okay, am I live? Okay, great. Let's see. This works. Okay, so I'm going to talk to you about where drug discovery is going, a little bit about my credentials. So as Howard said, I was 32 years at Pfizer, been working sort of independently for the last 10 years. The advantage of that is I'm not employed by anybody, which means no one looks at my slides and I can say anything I want. And so the trick here is to be honest and provocative without being too negative. Okay. So where is it going? And here's the outline of the talk. Okay, so I'm going to be talking about academic targets and a translational gap. Howard's last few slides nicely introduced that. And I'm going to raise this issue. Is it just a missing resource issue, or is there something fundamentally more of a problem? Then I'm going to be talking about chemistry and attrition, how it's growing worse with time. And really, is reductionism, the misapplication of genomics and high-throughput screening to blame? I know we have people later in the talk who are talking about high-throughput screening. I don't know whether they're in the audience here or not. Screening diverse compounds, that's commonly recommended. I actually think it's absolutely the worst possible way to discover a drug. I'll tell you that I believe the novelty drive comes from patents and not from the science. It's actually harmful to productivity in drug discovery. Talk a little bit about biology and chemistry network analysis and what the implications of that, and why chemistry due diligence on leads is absolutely essential. And then a final slide on what to look for. Okay. So what are the drivers for the drug discovery changes that everybody in this room almost certainly is aware of? Well, they really lie in three areas: chemistry, safety, and efficacy. Now, in the case of chemistry, so all of the kinds of considerations that you could see are related to the actual chemical structure of a drug. The predictivity is pretty good. So 2/3 of the cases, we have it completely under control. Okay. And here we have rules and filters, physical chemical properties, structural properties, and ADMEs, absorption, distribution, metabolism, excretion, productivity, worsens when you get outside of rule of 5 space. And I'm not going to explain rule of 5 space. So if there's somebody in here who doesn't know what it is, I'll be glad to explain it at the end of the talk or offline. Now with safety, it's not as good as chemistry, but it's still not that bad, about 50% successful predictivity. Its efficacy is the absolute killer. It's about 10% successful productivity. So more and more, as we go on in time, we realize that compounds are dying because of lack of efficacy. Now, if you were to multiply these out, 65% times 50% by 10%, you'd come up with a 3.5%, or 3.3% actually, success rate. Right? Now these figures are based on about 3 years ago. Last week I was in San Diego at a meeting, and the latest update is that 3.3 success rate has degenerated to 1.65%. That means you have to start effectively 66 drug discovery programs to get a successful compound out at the end, approved for marketing. Right? So it's really absolutely horrible. That's the only way to describe it. And this is one of the major drivers for using academic collaborations. So the idea goes is that the industry, people know this. They've known it for a decade. And it's getting worse. So what do you do in a situation where almost everything you try is going to fail? Well, one of the ideas is if systems biology were robust and advanced enough, that might be an approach. But it really isn't. So the fallback position is to say, well, where are the expert biologists? Well, they're overwhelmingly in academia, because that's where the NIH funding goes to. So maybe if you can tap into these pool of expert academic biologists, because they are experts, we might have a better success rate. That, in a sense, that's the argument. So the target quality is most likely from rich biology. And so this is a different cartoon of the Valley of Death. I think this is actually a little less interesting and a little bit more scary, right? But you definitely don't want to be stuck there on a hot day. And, you know, the gulf between biomedical research and discovery, Valley of Death, here's one of the earlier publications. There's like a ton of stuff on this. This is 2008. Everybody recognizes there's a problem. There's a problem in translating basic biomedical discoveries coming out of academia into something useful in drug discovery. So, now what are the causes of this? And again, there are all kinds of literature. And what I've tried to put together is what I consider to be the politically correct causes. Now, these are all correct. They're absolutely correct. But they don't create controversy. You know, so academics lack drug discovery skills. Okay. It requires industry-academic collaboration. Many of the groups that need to talk to each other are not correctly situated. For example, at least traditionally, medicinal chemists were mostly in industry. The biologists were mostly in academia. Now that's, of course, changing a little bit now because of the layoffs in pharma and trained pharma medicinal chemists going to industry. But that traditionally was a big problem. It still is to a considerable extent. You know, academics typically don't have access to the early preclinical properties, absorption, distribution, metabolism, excretion, early preclinical toxicity, don't have access to drug metabolism, pharmaceutical sciences. Why? Because these are generally not critical disciplines within academia. So there's no easy access. There's no access to preclinical and clinical interface skills. For example, analytical process chemistry formulation. Again, they're not part of the typical academic setup. Now, in the more modern era, they might be part of a core facility within an academic center. But traditionally, they haven't been part of an academic skill set that you can lay your hands on. And there's no access to early developmental skills. For example, toxicology, biomarkers, especially things like project management. I have a whole story, you know. Academics in general are pretty hopeless. I want to get something thrown at me, but pretty hopeless in project management. So it can be viewed as boring, but really in terms of true drug discovery and development, you really need to have that discipline. Okay, so these don't raise people's hackles. Okay, that's fine, you know, it's a problem. Now here, the thing that really does cause problems. Underlying sort of the academic industry collaboration or bridging the translational gap is the assumption that academic ideas on new targets are of high quality. And there was a landmark publication from Bayer last year. So the Bayer people reported on multiple hundreds of academic targets over the years that they had imported. And what they were seeing is that 50% of the academic targets were just out and out wrong, right? I mean, not just a little bit flawed, but out and out wrong. And another 25% were partially flawed. So there was something there, but it wasn't as good as it was advertised. So I think it's a reasonable assumption that the translational death valley exists in part because of poor quality academic target identification. Now, I actually raised this issue. So this is, what I'm saying here is not new. I actually raised it at a conference last week, and I really got shut down by an industry person who was a senior person in industry, who's right at the interface of this industry-academic collaboration, because this panel discussion was going out over 230 sites on the internet, and this is really politically charged. Okay. So what's the behind the problem? Well, I think, this is my opinion, the culprit is primarily the pressure to publish to support both grant applications and career development. Okay? It's a people problem and it's a government problem. Okay? And a people problem. Why? Well, because there's no malice here or dishonesty. It's natural human reaction. If you're a biologist, your career and your publications and your graduate students and postdocs depend on success in experiments supporting your hypothesis. And government comes in here because NIH overwhelmingly funds hypothesis-driven research. Well, guess what? There's a huge driver to find the set of conditions so that your hypothesis comes out correct. And it's not entirely a biology problem by any means, because the situation is compounded and embedded by a wealth of poor quality chemistry compounds. So it's the driver for success in an academic career, compounded by the easy access of poor quality compounds that will help support what is actually an incorrect hypothesis. Now, the good things about this is there are a lot of efforts underway on this. So if you read about academic industry collaborations, it seems like every 2 or 3 weeks there's something else reported. And this problem does get fixed at a late stage. It gets fixed because the VCs and the pharmaceutical companies have learned and gotten burned, and they won't invest significantly in a project without independent verification of what comes out of an academic lab. And I was last year at a conference, and shortly after the Bayer report came out, and the VC guy told me, he said, yeah, he said, Chris, you know, we'll spend £500,000 or £1 million to check out, independently verify in a laboratory what's been reported in the academic group before we seriously invest any money, because we've been burned so many times. And here's the Bayer observation. This was again last year. Okay, now the fact that the success rate has been the more analyses there are, the worse the success rate gets. I told you right now it's down. It takes 66 projects to get one out of the end. Has led people, this is so bad that it has led people to question every single step of the drug discovery process. And one of the steps that people have been questioning is, has in fact the nature of preclinical experimentation as applied to drug discovery, are there major flaws in that? Now, I would say certainly over the last 20 years, the prevailing mantra in drug discovery was identify a mechanism and screen against that mechanism to discover a selective ligand against a single target. Right? And what we now know is, first of all, at the very, very early level, for example, if you take a look at work in yeast, complete pathway knockouts, 85 to 90% are phenotypically silent. Nothing happens. The signaling network just goes around it. And then on the very other end, the clinical end, practically nothing is getting through, which suggests that although it is possible to discover a drug through a selective compound against a single mechanism, it's rare. If I had to pull a number out of the hat, I'd say it's no better than 10%. And it's also compounded by the fact that we now realize it's virtually impossible to find a drug that's selective. So in fact, there's probably a great deal of serendipity in clinical success, that there are aspects of drug action that we don't understand that add to efficacy. So the result of this is the whole range of counter responses. And when you read the literature now, you'll see these. So what are these responses? Well, one of them, Howard mentioned, phenotypic screening. The fact that when you screen for phenotype, and you're not, especially in a mechanistically unbiased scenario, you tremendously open up target opportunity space, because there could be literally hundreds of mechanisms that were responsible for the phenotype. If you screen for a mechanism, you impose your bias. You're only screening for one mechanism, and you could easily be wrong. Drug repurposing, really hot area. Again, you've probably been reading about it. Oh, the Alzheimer's area, a retinoid, targretin, just came up in the press a couple days ago. In animal models, potentially might be, these things are all hyped up, right? But it's an example of something you almost see every week or two. Multi-targeted drug discovery. Right? So because of what we know about network biology, it's very often the case that you can get a much more profound effect if you intercept a signaling pathway in a very modest sense at 2 steps, rather than having a complete knockout at a single step. It has to do with the construction of signaling networks. So why not deliberately try to incorporate more than one mechanism in a compound, or deliberately mix compounds together? And multi-targeted drug discovery, by the way, that is the pattern in CNS compounds. There's virtually nothing that works in the CNS area that is selective for a single mechanism. More of an effect, more of an emphasis on in vivo screening. So again, changing your assay so you go as close as possible to the clinical situation, and you get as far away from sort of the biochemical, excessively reductionist approach that has dominated a lot of work over the last 20 years. And finally, non-target, non-mechanism screening, so people actually looking at metabolite fluxes, or looking at effects along pathways. Now, this slide, I just point out that, at least to me, there might be a parallel between genomics and chemistry. So the genome sequence, the Science paper came out around 2000. Automated chemistry, so combinatorial chemistry, making chemistry the robot, started in 1992. And both